All right, in the last chapter, or last section, I guess we'd say, or part, we talked about the membrane transport. So here we'll talk about kind of a couple of the mechanisms that are involved with some membrane transport that's actually involving the production of vesicles. So if you recall some of the organelles that we learned about earlier in the year when we were talking about the cell, we talked about like the Golgi apparatus, we talked about the endoplasmic reticulum and so forth. Well, a lot of times what happens is these proteins get made into the endoplasmic reticulum and then they get moved to the Golgi apparatus where they're going to form some vesicles, and then they're kind of gotten exported out of the cell through the process of exocytosis. Okay, and that's where you get a vesicle fusing with the membrane and kind of dumping its contents into the surroundings. So the vesicle can contain waste, it can contain enzymes, it can contain any of a number of types of things. And again, it's just going to get this stuff out of the cell. So if we look here, here's a video that we can kind of look at and see how this works. All right. So looking here, make it a little bit larger for us. So you can see what you've got here is the cell that's being circled. This is going to be getting rid of some waste from this paramecium. It's just kind of a freshwater eukaryotic cell, but you can see there it's dumping it out right now out of that vesicle that's migrated with and fused to the plasma membrane, kind of dumped it out. Okay, so that's just an example of exocytosis. All right, so that's that. Now, looking here, if we continue on here with endocytosis, this is where the process is kind of reversed. So this is where the cell's going to fuse with some sort of an external type of an object, and kind of it's going to pinch inward and kind of bring a substance into the cell with a membrane wrapped around it. And now it's got a vesicle that it can kind of fuse with a lysosome and I guess take the contents of what it's brought in, break it down, and use it for energy if it needs to. Okay, and there's three main types of endocytosis. You have phagocytosis, pinocytosis, and receptor-mediated endocytosis. And with phagocytosis, what happens is the cell's basically going to take some pseudopods and kind of wrap it around the object, engulf it, and kind of bring it in, and then fuse it with a lysosome, which is a bag of enzymes, hydrolytic enzymes, and it's going to break the contents down of what it brought in. So it's the way that some organisms eat. So if we look here at this particular video, we can see the process of endocytosis. I'll make it a little bit larger here so we can see it. So this is an amoeba that's kind of extending its pseudopods outward. So if we're looking at it here, we can see the pseudopods kind of wrapping around this object that's been trapped, and it's going to basically form a vesicle, and then this object is going to, or the vesicle is going to fuse with the lysosome. The lysosome is going to dump some hydrolytic enzymes into these new vesicle that's formed, and it's going to break down the object that's in there. So that looks like it's probably a bacterium or something that's been trapped, and then it can't get away, and now basically the tonicity has changed inside the cell, and the cell no longer functions, and now this amoeba that's kind of engulfed this has a meal. Okay, so that is endocytosis. So if we go back and kind of look here now, we've got a lot of little videos here just kind of showing this same type of a thing. So here you'll be able to see what's going to happen with this type of an organism is it's going to shoot out some pseudopods, wrap it around the object, and kind of engulf it and bring it in. Okay, so I'll make this one a little larger for us. So you can see it's kind of got this object that's been trapped to it. The thing cannot get away, and pretty soon you're going to see these pseudopods kind of shoot out of it. As it's trying to get away, it's getting engulfed further and further, and then it forms this vesicle, and now it's got it trapped, brings it in, and then it's going to fuse it with a lysosome, and again digest it and use the contents for energy. Okay, so those are a couple of examples of endocytosis. Okay, now pinocytosis, I don't really have any videos here, but basically what you're doing is the outside kind of surrounding solution contains things that the cells need, so it's going to form these vesicles and kind of pinch inward and bring them in, and you can see all of the vesicles inside of here, that contain fluid from the surroundings and so forth. And that's just pinocytosis, which is cell drinking. Phagocytosis is cell eating. Okay. Now with receptor-mediated endocytosis, this is a way in which certain ligands or small objects in the surroundings are going to bind with certain receptors on the surface of the cell and bring these things in. And when this binding occurs, it's going to kind of initiate a series of events that's going to allow this vesicle to pinch inward and bring in the substances from the outside. Okay. So here's a video that will kind of overview the entire process of receptor-mediated endocytosis, and then I will explain it a little bit further to us as we go through this, okay?
Cholesterol circulates in the bloodstream and then enters cells by receptor-mediated endocytosis. Instead of circulating freely, cholesterol molecules are derivatized and packed inside low-density lipoprotein particles, or LDLs. A protein and phospholipid layer surrounds the cholesterol molecules. The protein portion is recognized by LDL receptors on the surface of cells. An adaptor molecule, called adaptin, binds to the tail of the LDL receptor that protrudes into the cytosol. Adaptin recruits clathrin molecules, which start coating the membrane. Assembly of the clathrin coat causes the membrane to bend and invaginate, forming a vesicle that buds off inside the cell, taking with it LDL receptors and the LDL particles bound to them. Once inside the cell, the vesicle uncoats and fuses with the endosome, the intracellular compartment that first receives all endocytosed material. The endosome has low internal pH, which causes the LDL receptors to release their cargo. Empty LDL receptors are recycled to the plasma membrane in vesicles that bud off from the endosome. Each LDL receptor makes a round trip from the plasma membrane to the endosome and back every 10 minutes. Meanwhile, the LDL particles need to be disassembled. The endosomal content is delivered to a lysosome, which contains hydrolytic enzymes that can digest the particles. Free cholesterol is liberated, together with amino acids and small peptides generated by digestion of LDL proteins. The cholesterol is then released into the cytosol to be used in the synthesis of new membranes.
All right, so that talks about receptor-mediated endocytosis. And the thing to kind of keep in mind as you go through this is that cholesterol example is used because during the development of these cholesterol drugs like Lipitor and things like that that'll help lower the cholesterol in people who have high cholesterol, a lot was learned about this process. So that's why these videos are containing stuff that has to do with cholesterol metabolism. Okay, so here's just a little figure that kind of goes through pinocytosis or cell drinking, phagocytosis, which is cell eating, and then receptor-mediated endocytosis, which is also a form. So these are the things that we've discussed so far and how things are kind of brought into the cell. Okay, so the process of endocytosis and exocytosis are ways we bring things into the cell and get rid of them and so forth. And it's also a way that the cell's plasma membrane is continually rejuvenated, because when you're forming these vesicles, when you exocytose something, that vesicle is going to become a part of the membrane, and when you endocytose something or bring it in, then when the vesicle pinches inward, you're going to kind of lose a little bit of that membrane. So that's a way that the cell can kind of build up and break down the membrane and kind of keep it new throughout these entire processes. Okay, so excuse me, this is a video that we're going to watch in class because it's about 15 minutes long. So we'll take a look at this one in class. But just as far as like going through receptor-mediated endocytosis now, the LDL or the low-density lipoproteins, these are the so-called bad cholesterol. And this, when you have your cholesterol checked, they're looking at LDLs and HDLs, but you want your LDL levels to be low. And the way that these things really are, I guess, stayed within an acceptable level in most people in their body is through this process of receptor-mediated endocytosis. Okay, and this is where the cell is going to bring these things into the cell and out of the bloodstream. And there's two mutations that kind of lead to either no uptake or a limited uptake of the LDLs from the blood, and then that leads to an increase in the amount in the blood and really, I guess, the symptoms associated with high cholesterol. So if we look at this figure here at the right, what we can see, and it just kind of reiterates the things that we talked about. And you don't really need to know all of the details. What you need to know really is that this cholesterol is going to bind with the receptors on the surface of the membrane. in what was called that clathrin-coated pit. So in other words, there's proteins that are involved in binding with these LDLs. You bring them in, and then you're basically going to fuse them with an endosome, dump the contents into the endosome, and then these receptors are going to be relocated back to the surface of the actual cell. So these things are just constantly being rejuvenated to the cell membrane without having to make more of these receptors. And then once those contents have been dumped into this endosome, we fuse it with this lysosome down here. And what that's going to do is break down the cholesterol and make it available for things in the cell. And the cell membrane contains some cholesterol. And then there's other kind of cells in the body that are responsible for making hormones out of the cholesterol and things like that. So cholesterol has a variety of uses, but we don't want it in our bloodstream, because what it's going to do is going to lead to the formation of plaques, and it's going to clog our arteries and so forth, and could lead to heart attack, stroke, things like that. Okay, so your body has a natural way of bringing these things in, and you want to keep your cholesterol, as low as possible, ideally under 200. And this is the natural way that the body actually does that. Okay, so when you're looking at this, what these things basically, I skipped over some of the things that I was just talking about, how this whole process worked when I walked us through this figure a second ago. And you can see here what this is it's a blown up region basically of this right here. Okay, so as we look at it, you can see the LDL cholesterol level, the LDL cholesterol molecules are going to bind with these LDL receptors. You have this clathrin-coated pit, which is going to invaginate inward and form a vesicle, and that's what's going to cause the cholesterol to be lowered. And in some of the people that have the mutations, you can't bind these receptors to that clathrin-coated pit, or you don't have any of the receptors at all, and that's what causes the cholesterol to remain really in the bloodstream, and bad things happen. Okay, so let's just look at this, and we'll kind of talk about and see how hypercholesterolemia, as it's called, high cholesterol, is kind of affects your body.
Familial hypercholesterolemia, or FH, is a genetic disorder characterized by greatly increased LDL levels and a family history of premature atherosclerosis. Normally, LDL receptors on liver cells help clear LDL particles from the bloodstream. FH may be caused by any of over 1,000 specific mutations that affect 3 known genes. More than 80% of these mutations increase LDL levels by causing the LDL receptors on hepatocytes to either be functionally altered or underexpressed, decreasing the body's ability to clear LDL from the bloodstream. The result of either scenario is an increased plasma concentration of LDL particles. In addition, impaired LDL uptake also decreases the cholesterol pool in the liver, which stimulates hepatic cholesterol synthesis and the production of VLDL, a precursor lipoprotein to LDL. These defects found in FH can result in an increased concentration of LDL particles starting early in life. Sustained exposure to high LDL levels and increased deposition of cholesterol into the artery walls over time elevate the risk of atherosclerosis and premature cardiovascular disease associated with FH, making early detection of this genetic disorder of critical importance.
And what atherosclerosis is basically hardening of the arteries and the formation of the plaques and clogs that you get in these arteries. Okay, so that's basically kind of in a nutshell what receptor-mediated endocytosis is and how it's kind of important to us. Okay, so that kind of wraps up everything with Chapter 7. So again, as always, make sure that you write down your questions, and then we'll talk about them in class, and we'll talk to you soon.